advertisement. If you're looking for graduate programs, yeah, it's just that city. Don't be that city. Whatever, it's my philosophy. Open house coming up end of this month, as you can see. If you're interested, I can send this to you or take a snapshot. Yeah, I can send this. Yeah. I was asked to circulate this amongst classes, so here it is. If you're interested in one of these degrees or one of these fields, go to North Campus there and have a meeting with current grad students and food and fun. So share the word. Be there if you're interested. Okay. Exam, right? Next exam's a week from today. Do review session Tuesday. I'll have a Zoom link posted by this weekend, or sometime this weekend. Yeah, I think I have other announcements. Any questions or comments on stuff before jump into today? Okay. All right, so today we're doing the one lecture on performance enhancing drugs. So as the name indicates, these will help your performance primarily with athletics, but with others, some with some other activities. But for the most part, or some of these will help. I guess if you're trying to get ripped or jacked or swole, is that swole still in circulation now? So for that kind of stuff, but a little roadmap of where we're going to go, or the specific ones. So the anabolic steroids that are good for that, growth hormone that are all kinds of performance features that'll help you with, erythropoietin, kind of a special drug that's, well, we'll get into that. Yeah, for boosting your oxygen content. Amyl nitrite, just one quick little blurb on that, as that helps with after hours performance, I guess, but I should have formatted this better. We'll just do an overview, pharmacology specific details for all of these. Yeah. And I guess a quick one slide review at the end. But diving in, anabolic androgenic steroids, or AAS. So these are the type of drugs that mimic the effect of testosterone in a system, in anyone's system really. And without going into all of that system, that's just what they do. These drugs are administered through pills or through intramuscular injection. Is absorbed fairly slowly within an hour or two, and then large first pass metabolism with the oral administration. But kind of the basis of how these work, by looking at this figure over here on the left, I need to put a label in here. So on the left is sort of the standard view of testosterone in circulation in someone, in a normal individual, while using anabolic steroids. This is their the breakdown of this actual testosterone. So T stands for testosterone here, but albumin buying testosterone, about 50% of the testosterone is bound by albumin in normal situation, 2% of it's free. Then CBG and SHBG are just circulating proteins that also bind to the rest of the testosterone, okay? But the idea here is that the bioavailable testosterone, so the free and then that which is weakly bound to albumin, are available. About 50% of total testosterone in the system is available for use in a normal situation. Now you skew that ratio if you start to deliver these AAS substances. As you can see, there's like a 50% increase of the albumin bound, same amount of free testosterone, and then that just cuts into the tighter bound testosterone with the CBG or SHBG proteins, all right? So the idea here is that when you start using these AAS compounds, it will increase the amount of bioavailable testosterone by roughly 50%. And yeah, this small increase here in the free testosterone actually works out to be quite a big difference in the performance of this molecule, or the ability for this molecule to influence the system. So that's the biggest, or that's, I guess, the first improvement that happens when taking these types of drugs, is that you increase the bioavailable testosterone by roughly 50%. So again, these mimic the effects of normal steroids. Testosterone, 5-dihydrotestosterone are the main active ones, with testosterone being the one most important. So the complementary synthetic pathway for this molecule, cholesterol, is the precursor to all of these. Don't need to know structures and names and all this. I show this just because this is where it comes from, but again, this is in biochem, so you don't need to know all these. Just know that, I mean, it's a fairly involved pathway to get to cholesterol into progesterone as well as testosterone, and then the DHT, or the dihydrotestosterone. So these are the main ones we're interested in here. But as you can see from the picture on the right, the synthetic steroids come in several different forms. But obviously they have that same 4-ring structure that comes from cholesterol, and so that's how they're able to bind to the receptors and have the activity that they do. So very straightforward there. Now the way that they work is through nuclear receptors. Nuclear receptors is the one class that we haven't covered so far in this class, but they're only applicable to these drugs. And the way that they work is something like this. So with the endogenous ligand or with an exogenous AAS molecule, it gets into a cell, right? So here's our cell membrane, outside, inside, in the cytoplasm. The receptors, these nuclear receptors, are bound to inhibitors prior to activation. Bound to these HSP. So heat shock protein, you just need to know HSP, but the individual monomers of this receptor are bound to an inhibitor, HSP, prior to being bound by an activating molecule, or endogenous or exogenous ligand. What happens once this molecule binds, so it kicks off the inhibitor, but it also causes a dimerization event of this molecule. Once the receptors are dimerized, they're able to translocate into the nucleus and have their effect there by, once again, affecting transcription of the cell, and then certain other changes we'll get into in a minute. I just want you to know this part. Don't worry about all of this other activity, the CYPs and the mitochondria and the this is and the that. Don't worry about that. Just know that AAS molecules bind to nuclear receptors, displacing the inhibitors, causing dimerization of the receptors, and the receptors then translocate to the nucleus, where they will affect transcriptional events, and then all the downstream fun that happens after that. So these work as agonists at the androgen receptor, or AR here. So a little bit more, so same thing we just looked at with a bit more players involved, or some names involved. Mechanism of action here. So intracellular glucocorticoid receptor specifically is the one that's being targeted. Now, endogenous ligands for this receptor are glucocorticoids and androgens, right? And depending on what binds to these receptors will determine what happens in the cell. So the AAS molecules are competing with glucocorticoid for control of muscle mass, is one of the biggest influences it has, right? So as we just covered, we've got these molecules, so androgens are in red, glucocorticoids are in green. They can bind to the receptor, the monomeric unit, displace the heat shock protein or HSP inhibitor, cause dimerization. This should be on the outside of the nucleus here. Dimerization happens out here, it allows it to translocate, and then will bind and cause a change in transcriptional activity. But here's a bigger outcome, or more important outcome. Amino acid degradation, nitrogen excretion, and reduction of muscle mass. Make note that this figure is showing what happens when glucocorticoids are bound. Okay, you see the green circles here. This is what happens when glucocorticoids are bound. When AAS are bound, the opposite of this happens. So these steroids change to a negative nitrogen balance to positive, all right? So this nitrogen excretion will then be nitrogen accumulation. Reduction of muscle mass will then be increase in muscle mass. And this will enhance protein synthesis in skeletal muscle, okay? So when the androgenic steroids, exogenous agents are bound to the receptor, they cause a switch from negative nitrogen balance to positive, enhances protein synthesis in skeletal muscle, and causes an increase in overall muscle mass, contrary to what glucocorticoid endogenous ligands binding does, right? So interestingly enough though, early studies with this really showed that there wasn't a lot of efficacy in this space or with these molecules. However, as it said, a lot of the early studies didn't use the supraphysiological doses that are used in performance enhancement. When they started doing studies on those, one of the biggest distinctions they found is you need to be an actively training athlete, actively training person in order for these to have an effect, right? You can't just take these steroids and sit around and get big muscles. You have to be training actively, okay? And intensely, really. You really got to be going after it for these to really have a big noticeable effect. So yes, for the most part, yes, not only do you have to be actively training, but you need these supraphysiological doses for them to have an effect, is the biggest bottom line with these drugs. However, problems arise with these drugs due to various reasons. One of the biggest are the behavioral effects. I guess roid rage is the common colloquialism that is used for people that turn very aggressive when under the effects of these agents. And yeah, there's a considerable amount of work trying to understand exactly why that happens. Some of it is very apparent why it does, but that's one of the effects. In terms of actual physiological effects though, this can affect how your circulatory system operates, especially in terms of thrombosis or development of blood clots that happen, as well as erythrocytosis, so lysing of red blood cells happens. Hepatotoxicity is an issue, especially in development of teratogenic effects, so development of tumors there. Shrinking of testicles is a big, a common issue and problem, and one of the, one of the aspects that's more comical or looked at in this field. Those being subjected to it don't think so. But then on top of that, musculoskeletal injuries are very big too. And it really is, I guess, very straightforward why this happens. Due to the fact that someone's body will become stronger and more massive before the rest of the body is able to acclimate to that is a big issue, right? Commonly when someone trains, especially when they're going to put on mass, if they're not using anything, the body has a very natural progression towards getting larger. And it will do it in a way that maintains an increasing, it'll stay at a certain homeostasis, but all of the systems in the body will increase as this person is putting on muscle mass, right? When you sort of give it this unnatural boost and you just start to develop more mass and strength outside of things, that's when you have a lot of problems, because people can actually not only tear their tendons, but they'll rip that attachment right off of the bone just because they become too strong for their own system before the rest of the system catches up. So bone ruptures and tendon ruptures and those sorts of things are, I don't know how common, but they definitely happen enough to be a standard sort of adverse effect with these drugs. All right, moving on to growth hormone. So sort of similar, but different, because this doesn't provide so much, yeah, it'll enhance your performance, but it isn't so much about bulking up and increasing strength. It'll actually strengthen the muscles you have and cause them to have better performance, being more lean, as well as helping with recovery after exercise. And there's a lot of interest in this field right now, just due to the fact that it doesn't seem to have, you can boost your performance and help your recovery after intense exercise, but it doesn't seem to have as bad an adverse effects as the AAS type drugs do. But what's important here, or way this is, excuse me, the standard growth hormone pathway works, excuse me. So, in the hypothalamus, we have growth hormone releasing hormone, GHRH. Don't worry about SST. That gets released by the hypothalamus, will act on the anterior pituitary and cause production and release of growth hormone, all right? Growth hormone can then be released, and it acts on a number of target tissues, causing those tissues to then produce IGF, insulin-like growth factor 1, just no IGF. And then that acts on secondary tissues, okay? So for this pathway, just know GHRH, or the hypothalamus is stimulated to produce GHRH, that acts on the anterior pituitary to produce and release growth hormone. That acts on multiple target tissues. Those then produce IGF-1, and then that has its effect on other tissues in the body to help with this performance enhancement, if you will. So, growth hormone acts as an agonist at its specific receptor. So these growth hormone receptors are this class of receptor we didn't talk about before, the receptors with intrinsic or associated kinase activity, but that doesn't matter so much. Just know that a monomer, so similar as our nuclear receptors, except for these are embedded in the membrane again. Once growth hormone comes in and binds, it'll cause the dimerization of two monomers, so you have a dimeric receptor here. And then once that is dimerized, and through this a series of phosphorylation events will trigger a bunch of effector proteins to cause expression of the IGF-1 gene, or insulin-like growth factor 1. So, binding of growth hormone to its receptor, acting as an agonist, cause dimerization of the receptor, which then in turn causes production of IGF-1 to increase, and then IGF-1 goes into circulation and will help stimulate a number of effects, most notably production of lean muscle mass through lipolysis, or breaking up of fatty acids. So, and this is the, main activity here. So, growth hormone and IGF-1 stimulate lipolysis. So it's a fatty acid breakdown, but it will help muscle mass become more lean. It'll cut down the storage of the fatty acids in muscle and stimulate glucose storage in the liver, so that you have larger stores of glucose for performance. While performing. So this helps for, it'll increase protein synthesis specifically in muscle and then other target tissues, but allows for muscle recovery and then bone growth in certain situations. But the big effect is the lean muscle development through lipolysis, and then the energetics of glucose storage, and then recovery through having more more glucose available for performance as well as recovery afterwards. And one last note on this, so recombinant growth hormone has one specific form. So 22 kilodalton protein is made. And so native or endogenous growth hormone that's produced has a lot of post-translational modifications to it. So if you actually do an analysis on growth hormone from an individual, you'll see there's a number of different sizes for that growth hormone. When agencies that are testing for enhancing supplements, if they see someone whose specific growth hormone profile has a large amount at this specific molecular weight that looks like someone who is trying to, who is using this sort of enhancement agent, just due to the fact that the exogenously produced growth hormone usually just has that one size to it, whereas endogenous growth hormone has an array of different sizes due to the post-translational modifications in our body. So, one detail on that. All right. How does that sound? Or questions? Anything hopefully straightforward with this one? All right, on to erythropoietin, or EPO. This is an agonist at erythropoietin receptor in early red blood cells. So looking at hematopoietic cell lineage, again, you don't need to know this whole thing, I'm just showing it for background, but you have a multipotent hematopoietic stem cell here. A number of factors will cause it to undergo different forms of differentiation. At one point, it will start to express the erythropoietin receptor. If you apply erythropoietin to this cell, it would cause it to develop into an erythrocyte. Final maturation as a red blood cell or an erythrocyte, okay? So, just working through the normal receptor, it causes a stem cell of a bloodline lineage to mature into a red blood cell. And then this effect causes an increase in the red blood cell count. And as I hope everybody knows, what red blood cells do in our body is carry oxygen and deliver it to muscle cells. So hopefully it's understandable that if you have more of these agents in circulation, you can have a higher amount of oxygen being delivered to muscles, and this helps for performance, especially in specific types of more endurance style sports, I guess, or the aerobic style sports. Cycling is a big one, and we'll get into that next here. So, previously what people used to do in sports is that, or before this was developed, they would do blood doping. Maybe I do have that on the next slide, yes. Or blood packing, and what people would do is actually draw blood. You know, they would have a race coming up, and several months beforehand they would take a pint of blood out and then spin it down, stick it in the freezer. And then a week or two later they'd take another pint of blood out and they'd spin it down and stick it in the freezer. They'd do that several times, and then they would condense the blood down to just the red blood cells, or they'd isolate it. And then the day before a race, or right before a race, they would have that reintroduced back into their system, and they would boost their red blood cell count that way, okay? And it'll just give your body the ability to carry more oxygen to muscles, helps you perform as well as it helps you recover. This protein was initially developed by Amgen as epoetin alpha, that's the recombinant form of this, used to treat anemia and then red blood cell loss during chemotherapy. So this is actually a cancer drug, or a drug for anemia, but then, you know, obviously when people started to see what it actually does, and it helps to, you know, in normal individuals who are not anemic, it can actually boost your hematocrit levels, or your amount of red blood cell in circulation. And so people started picking this up again. As listed here, those are involved in aerobic sports. And But this replaces that blood doping or blood packing that used to be done, because you can just take a shot of this and then your red blood cell count goes up. So one last detail on this. So similar to growth hormone, erythropoietin, endogenously produced EPO, has post-translational modification. So this top left panel is the primary structure for erythropoietin. As you can see, all of these circles with letters in it, that's the one or the single letter abbreviation for the amino acids. So this is just the long polypeptide chain of erythropoietin. But what you can see at various residues here are these branched kind of structures or oligosaccharides that are attached to the EPO protein. And it does this in order to increase the length of circulation for this protein, right? So if you can imagine, you got a protein that's going around in circulation. There's also a lot of proteases that go around, and it can clip up proteins just at random. But if you have these using oligosaccharides here, so just units of sugars, multiple sugars stacked together, that can actually fend off proteases so that the protein doesn't sit in the active site of those enzymes and get chopped up. So it's just a way of increasing the lifetime circulation for proteins. It's very common throughout mammals, well, throughout all of them, I guess, chordates really. In humans is all we need to worry about. So over on the right then, this is just a schematic of the glycosylation patterns, because what Amgen did, after they saw that this is the endogenous kind of sequence for erythropoietin, they figured, you know, if EPO lasts X amount of time in circulation as is, what we can do is add more glycosylation sites, increase the post-translational modification, and then hopefully increase the amount of circulation for EPO even more. And that's what they did. So here is just a basic schematic of exactly this, with these different oligosaccharides shown here, and then the smaller ones here, I guess, is here. The next generation of this molecule is called darbepoetin alpha, and that really just has more glycosylation sites on this structure, okay? And so what this allowed for is it further protects the protein, so it lasts longer in circulation, it has a longer half-life or activity. And then, because they weren't satisfied with that, they developed another form of EPOGEN. So here again is endogenous EPOetin that has these glycosylation sites on it, but what they did is create this SERA molecule, continuous erythropoietin receptor activator. So this is a modified EPO that has lower affinity, but it prolongs the effects here. And so what they did is not only do they have, so here's our EPO here, here's our modifications. This great big 30 kilodalton chain is polyethylene glycol, which is kind of a neutral substance that can increase solubility of proteins in circulation. But the biggest thing again is that it really fends off proteases and things like that. So it allows for protection of the molecule, and this SERA is the longest acting EPO that we have that's available. So again, the idea is you can protect proteins with post-translational modifications. Our bodies do this naturally. You can also engineer these into proteins so that they last even longer in circulation. And that's this first generation here, where they have more glycosylation sites, they have more post-translational modifications on this protein, and allows for longer circulation. And then building off of that, but instead of just using these oligosaccharides, they added this huge chain of PEG to this protein, as shown here. So this is even bigger than the endogenous protein itself. Actually, that's not true. Yeah, it's right the same level. We're at 30 kD for epotin, 30 kD for the PEG chain. But the idea is that you're adding bulky things to a protein that allows it to stay in circulation longer, so you have a longer activity from this protein. Alright, so the last bit on that, the U.S. Anti-Doping Agency, or USADA, came out with the Clean Sport Handbook just this year. You can look this up. And it's, what it is obvious, and it just says, don't do this and don't do that if you want to be a professional athlete. And you can look it up at their site there, but it gives guidelines for what you can do and what you cannot do, what's considered performance enhancement in a legal way or an acceptable way, and then what's illegal or unacceptable in terms of performance enhancing substances. So, all right. So, quick aside on amyl nitrite. These drugs, also, colloquially known as poppers. This is just a molecule that increases the nitric oxide in solution or in circulation, acting as a potent vasodilator. So it just allows for the vasculature to swell more, allowing for greater circulation in certain areas. These primarily they're medically prescribed for angina, so chest pain, not necessarily associated with physical exertion, but sometimes it does, and especially popular in those with CHF or chronic heart failure. But this drug is used to treat that as it rids them of pain due to the fact that a lot of times they have constricted arterioles due to buildup of plaques and other bad stuff in circulation. So this will allow for relaxation of the vasculature and better circulation. It also works in cerebral blood flow, so in the brain this can have an effect and gives warm, fuzzy feelings, and also heightened sexual arousal. And before all of these fun things that have been around for the last 20 years, the sildenafils and the Viagras and the whatnots, this was the one that people used to go to back in the 70s and whatnot for their Friday night activity. Kind of phased out. I don't hear people using these as much as the others anymore. But so that's it for performance enhancing drugs. Just a quick review, I guess, for this next exam. This will cover lectures 7 through 14. The alcohol and benzodiazepine barbiturate lectures, both the marijuana or cannabis lectures, a couple on opiates, and then the one on performance enhancing drugs. Tuesday we've got media in the illicit drug use space, if you will. I'll tell you there what's going to be important for the exam, but for the most part, again, it's going to focus on the drugs of abuse. But I did want to highlight the in vivo experiments that were being covered in this section. Standard discrimination protocol, so just feeding a rodent some drug in sugar water versus just a sugar water control and having them discriminate between the two. That was an experiment we covered earlier. Elevated plus maize and novelty suppressed feeding are both for anxiety, studying anxiety. Standard discrimination it can be for anything, any type of drug. If you want to see if it has some sort of desirable, those are the only things we've talked about are the desirable drugs. There's also punishing drugs that you want to stay away from that will cause the mice to not use, but we didn't get into that. Morris water maze is for memory, studying memory, if you remember that, and then the tail lift and the hot plate assays are for analgesia, or measuring pain in an individual. So please know those experiments, what they're used for, what basic data from them will be, and I think that's it. So hope everybody has a good weekend. See you on Tuesday. And let me know if you have questions.